dendrimers. They're about the size of molecules. So for instance, the most common one used is G5 dendrimers. And I'll just point out that here they're circular, but now people are starting to make them asymmetric, so that one side's kind of the targeting side and the other side's the payload side. But these can be coupled with a variety of things, for instance, metals like iron, gold, gadolinium, and they can be coupled to drugs. So methotrexate folate dendrimers were in clinical trials. They weren't very successful, but they've been there. This is an example of using a dendrimer, G5 dendrimer, with an iron oxide, and you can see the controls have a certain level of signal, and those with the iron oxide have a little bit lower signal. Another thing is the takeover of nature's known pathways. Now, there's one whole field called transcytosis, where the body transports particles and small molecules from the endothelial lumen to the abluminal side. But we won't talk about that. But there are other mechanisms that are important, and this is one, for instance, associated with HDL. HDL is a lipoprotein, the good lipoprotein that we all talk about, and it has the ability to bind to certain receptors called the scavenger receptor. And when you put a payload in the center of this, it's able to deliver that payload the same way it delivers cholesterol. And that's a delivery that does not require the particle going in the cell or going into endosomes and then having to break out. It literally injects their payloads right into the cell, much like a virus injects its DNA or RNA right into a cell. So this is a very hot area of research by about 3 or 4 groups right now to try to use nature's method of injecting payloads into cells so they don't have to have endosomal escape. This work came out of Toronto from Gang Zheng.